experience in studying cell-signaling pathways and drug discovery, took 2 drugs to clinical trials at the Institute of Cancer Research as a lead biologist. One of the drugs got orphan status from FDA, working as an associate professor at LUMS since 2014, where he has established cancer therapeutics lab focusing on early phase drug discovery and drug resistance. I would like to invite him to chair this session. Thank you. Now I would like to invite Dr. Suhail Qureshi, who is a molecular biologist with over 35 years of experience of PhD and postdoctoral experience. After receiving his PhD in 1990, he carried out his postdoctoral training at Glasgow University Imperial Cancer Research Fund UK and Cambridge University. Subsequently, he went to serve University of Wisconsin Madison as an assistant professor and Invitrogen Corporation as senior scientist before returning to Pakistan in 2004. Until 2011, Dr. Qureshi was at the Aga Khan University Hospital and thereafter served the Syed Babar Ali School of Science and Engineering at Lahore University of Management Sciences, LUMS. As a professor and dean, in 2016, he joined Seacoast University as a professor at the Institute of Integrative Biosciences and subsequently served as a director and vice chancellor. In January 2022, he joined the Department of Life Sciences, University of Management and Technology, UMT. Since 2024, Dr. Qureshi has been with Shalimar Institute of Health Sciences, where he is Director of Research and Innovation Laboratory. Dr. Qureshi has been the recipient of the Outstanding Teacher Award from Aga Khan University. His research interests include gene regulation in extremophiles, cancer, molecular evolution, biology of hepatitis C virus, and phage therapy. I would like to invite Dr. Qureshi to chair the session as well. Before I hand over the session to the 2 chairs, I would like to thank Dr. Sumayya Nizamuddin, who has been instrumental and a co-moderator with me to organize this session. So Dr. Suhail and Dr. Amir, the floor is yours to invite the guests. Thank you.
Okay, Bismillahirrahmanirrahim. Assalamu alaikum, everyone. It's my pleasure to chair this session along with Amir Faisal. And we're delighted. This is going to be, I think, one of the best sessions you'll find. I like to brag about this session because the lineup of speakers is just outstanding. And so I'm sure that, by the time we finish the session, there's a whole lot of learning that will have gone on in this room. And so the first talk is by, if I can have this, is it's by Dr. Hani Reddy. Sorry, what a time I'm taking adjusting here. And the title of her talk is Using Cerebrospinal Fluid to Inform Diagnosis of Central Nervous System Tumors. Dr. Reddy is a very experienced academic. She's currently serves as a senior vice president and medical director for Belay Diagnostics. And prior to her current position, Dr. Reddy was a professor in chief of the Division of Clinical Genomics at the Medical College of Wisconsin. With a background in biotechnology and specialized training in clinical molecular genetics from Mayo Clinic, she's also led a translational research program on thyroid cancer. And then her accomplishments go on and on and on. And I think it's time that we leave the floor to Dr. Reddy. So Dr. Reddy, can you hear me? Are you there? Virtually? Okay, so let's start her talk. Thank you.
Greetings, everyone. I'd like to start and the organizers for the opportunity to present our most recent work on cerebrospinal fluid liquid biopsy to inform diagnosis and management of CNS tumors. I'm Hani Reddy. I serve as the Chief Lab Officer for Belay Diagnostics. By way of introduction, I'm a board certified, ABMGG board certified molecular geneticist and experience in clinical and translational genomics. From a conflict of interest perspective, I'm an employee of Belay Diagnostics with salary and options. I also have a disclosure. This presentation is for informational and educational purposes only. The views, thoughts, and opinions expressed herein solely belong to me and do not necessarily reflect the official policy or position of Belay Diagnostics. The content does not constitute legal, medical, or professional advice. I'd like to start off with an outline of the presentation. So a couple of things, touching on a little bit of background for liquid biopsy, focusing on CSF or cerebrospinal fluid liquid biopsy for CNS tumors. And then I'll end with a couple of offerings that Belay Diagnostic has for CSF liquid biopsy to inform diagnosis and management of CNS tumors. So starting off with what tissue biopsy is, which is the standard of care. As we all know, it is invasive, takes longer, higher costs, obviously, to obtain the specimens. Molecular information is limited to the areas that are evaluated. It's not compatible with tumor monitoring, particularly during surveillance treatment. And this is where liquid biopsy actually comes in for cancer. It's a minimally invasive method to be able to evaluate circulating biomarkers. As we look at it from the cost effectiveness, the shorter turnaround time it takes, and the flexibility it offers to be able to look at complex tumor molecular information. But most importantly, it's also compatible with tumor monitoring during surveillance treatment. So there are a number of biological specimens that are currently used for liquid biopsy. There's blood. Everyone's very familiar with that. So you can have either plasma or serum that's used for liquid biopsy. There's CSF or cerebrospinal fluid that I'll talk a little bit more about. Urine to be able to track bladder cancers. Ascites for abdominal cancers, such as ovarian. Stool to be able to track cancers such as colorectal carcinomas, bone marrow, saliva, as well as pleural effusions to be able to evaluate lung cancers. So within this context, having so many different biological specimen types that can be used, there are a multitude of analytes that can be looked at. You can look at immune cells, you can look at proteins, circulating tumor cells or CTCs, vesicles, RNA, DNA, as well as circulating tumor DNA as well. So let's look a little bit about, as I mentioned, the different specimen types and the various cancers. Blood, plasma, and serum usually used for all cancer types. Cerebrospinal fluid, primary and metastatic brain tumors, more so because of the blood-brain barrier that does not allow plasma to be as sensitive as CSF. Saliva and sputum for head and neck cancer, pleural effusions for lung cancer, urine, as I mentioned, for bladder cancer, but also for renal and prostate. And then stool for colorectal and pancreatic cancer, bone marrow for breast, prostate, lung, and colon cancer. And then ascites for abdominal cancers. So just touching a little bit on the applications of liquid biopsy, as I mentioned, tumor is invasive. It is associated with, or resection of tumor is invasive. It's associated with comorbidities, but liquid biopsy is indeed minimally invasive. So it can be used for diagnosis and prognostication, assessment of treatment response, detection of resistance mutations, evaluation of recurrence, monitoring of minimal residual disease, as well as screening and early detection. So when you take a look at the various Specimen types, we just looked at. Plasma is the preferred specimen type over serum for liquid biopsy, or compared to other specimen types as well, depending on the kind of cancers you're looking for in terms of primary tissue origin. The reason plasma is preferred over serum is because cfDNA from serum gets contaminated by blood cells. The amount of contamination increases with the number of draws, as well as with the number of days between blood withdrawal and serum separation. So it's very important that you get serum out as soon as possible post blood draw. Serum is also associated with a higher false positive rate. So if you take a look at the different analytes evaluated in liquid biopsy, as I mentioned, you can have DNA, RNA, protein. You have extracellular vesicles, such as microvesicles, exosomes, and apoptotic bodies. They contain both DNA and RNA. You can have circulating tumor cells, as well as circulating nucleic acids. Could be cell-free DNA, cell-free RNA, or a circulating tumor DNA as well. So within all of this, different analytes that are feasible, cell-free DNA or circulating tumor DNA is the most preferred analyte. Reason being that it's easier to isolate than circulating tumor cells. DNA is more stable, obviously, than cells or RNA. The proportion of ctDNA, for the most part, is reflective of the tumor burden. Additionally, cfDNA has higher sensitivity and dynamic range. So that allows you to have a better assay when you're looking at cell-free DNA versus trying to solely depend on circulating tumor cells. However, every assay that one develops for clinical use does have its limitations. So there is limited sensitivity if prior information of a targeted variant is not available. So if you're doing liquid biopsy as a follow-up to tumor profiling, then your sensitivity is obviously increased because you know what you're looking for, and the liquid biopsy assay is now tumor informed. Not all variants can be detected due to tumor heterogeneity, so it depends on which portion of the tumor has shed and what DNA is isolated. There's also the limitation of available material. So, NGS has a detection rate of about 1.5%, about 1 to 5% variant allelic frequency. So anything lesser than that does end up being a challenge. It's also difficult to distinguish whether cell-free DNA is from tumor shedding, that is resistance, or tumor dying, which is apoptotic cells due to therapy, and therefore making sure that you use a decent number of true negatives or samples that are normal as part of your clinical validation, analytical validation is very critical to determining the sensitivity and specificity of the assay. There are various technologies for evaluation of cfDNA, but it does require very, very high sensitive techniques because of the small fraction of tumor-specific DNA that's mixed with wild-type cell-free DNA that is obtained from your liquid biopsy specimen. NGS is currently being used very widely for liquid biopsy assays to develop targeted panels. So it could be one gene, few variants, or a comprehensive genomic profiling assay, as I'll show a little bit further along in the presentation. This also enables high throughput, low cost sequencing to identify alterations genome-wide. As I mentioned, every methodology platform has its limitations. And so if you take a look at the limitations of liquid biopsy for cancer, it's difficult to distinguish germline from somatic variants. And why is this so? So when we do tumor resections, we can do a plasma or a blood versus tumor to be able to identify those germline variants. However, in liquid biopsy, you're not able to do that. Anatomic location of the tumor is often unknown. So from a screening perspective, or trying to understand where, for example, if you're doing blood, trying to understand where the actual tumor is becomes a little bit of a challenge. False negatives are indeed seen a lot more with liquid biopsy than with tumor because of the low threshold of detection. So this is indeed a challenge for early detection and minimal residual disease. Additionally, you could have missing variants due to tumor heterogeneity. False positives are another challenge with liquid biopsy, more so with CHIP or clonal hematopoiesis of indeterminate potential. So CHIP is actually defined by the presence of age-dependent acquired mutations in hematopoietic progenitor cells, and it's seen, has been reported to occur in about 30% of individuals 60 to 70 years of age. And so when you actually see variants in certain genes such as TP53 or PTEN, one always needs to make sure that it is not CHIP and is indeed a true positive. And taking all of this into consideration, when you're developing your assay, you need to be focused on a multitude of factors that ensure you have the right sensitivity and specificity. So the specimen type, the analyte to be tested, the nucleic acid to be evaluated, the type of variant you're looking to evaluate, whether it's simple or complex, the technology to be used, and then the purpose of the assay that ultimately drives your end product is whether you're looking for evaluation of response, resistance, or recurrence versus screening for MRD or early detection. So having kind of gone through the basics or the background to liquid biopsy, I'll start off with cerebrospinal fluid as a specimen type for liquid biopsy of central nervous system tumors. And this is something that our lab has been working on for the last couple of years, drawing from the work of Dr. Batagora at Johns Hopkins, who's spent more than a decade working on this. So let's talk a little bit about the diagnosis of CNS tumors. Approximately 25,000 individuals were diagnosed with a primary tumor. However, these numbers increase tremendously when you're looking at metastatic or secondary tumors, as well as those that are recurring. The 5-year survival for these is about 36%, which is one of the lowest across different cancers. Current treatment options for brain tumors include immunotherapy, surgery, which is extremely invasive, chemotherapy, targeted therapy, or radiation therapy. And within this context, you also see that diagnostic delay of CNS tumors is associated with worse neurological outcomes, in that 39 days on average, 26 days for high-grade tumors, and up to 138 days for low-grade tumors. And that is because the current standard of care for diagnosis of CNS tumors is threefold. First is, if you have common symptoms of a brain tumor, such as headache, vomiting, nausea, seizures, cognitive challenges, speech challenges, vision issues, etc. Basically, you can get, they start off with an MRI or a CT scan. They do a lumbar puncture for CSF cytology, and then ultimately go into biopsy, right? Which is, again, very, very invasive. Now, the MRI or CT scan can be non-specific because you could get something as simple as an abnormal brain scan that you really don't understand what it is about. It could be infectious disease, it could be inflammation, it could be demyelination, or it could be suspicious for a tumor. Lumbar puncture for CSF cytology has an extremely low sensitivity, about 41%, whereas biopsy, on the other hand, has a non-diagnostic rate of about 17% and a diagnostic accuracy that varies between 73 to 97%. So clearly, if you look at all the options currently available, the chances of being able to detect a CNS tumor in the very early stages are challenging. So this actually becomes a lot more significant when you look at the context of doing invasive surgery. So if you have to do a solid tumor biopsy, it allows for histological diagnosis, but then it's extremely invasive, a risky procedure, and sometimes it's not feasible due to the tumor and atypical location, and it's also not representative of the tumor heterogeneity. Whereas, as I mentioned, liquid biopsy of CSF now allows for us to kind of address these different aspects of limitations with solid tumor biopsies, with solid tumor biopsy or resection. However, the downside of liquid biopsy is it does not allow you to do histological characterization. There's a lack of standardization because you could do a lumbar puncture, you could use a Omeyer reservoir. There are so many different ways in which you can get access to the CSF fluid with some amount of limited sensitivity as well. So recently, the Blood Profiling Atlas in Cancer, the Blood Pack Consortium, came up with some recommendations and observations for CSF, identifying it as a more promising biofluid for liquid biopsy because the circulating tumor DNA content is orders of magnitude higher in the CSF compared to plasma and has been shown to be molecularly faithful to the tumor. Additionally, liquid biopsy in brain cancer has multiple use cases along the patient journey. And while a lot more work has to be done, there's definitely a lot of complementary uses of this technology in the context of the current standard of care. So if you take a look at the potential for CSF to inform diagnosis of CNS tumors, it's secreted into the ventricles of the brain, it flows in and around, can be obtained by lumbar puncture, and it's typically low in WBC, so those contamination issues that we are concerned about don't really come into play. These are the NCCN guidelines that very specifically talk about having next generation sequencing in CSF, and that it's important in being able to inform the diagnosis of CNS tumors. So along these lines, Belay Diagnostics developed two different tests. One is called the Summit, which looks at SNVs, MNVs, and indels, as well as chromosomal arm level aneuploidy, and Vantage, which looks at MGMT promoter methylation. So within our ClearCap lab, both these tests are validated. Our analytical validation and clinical validation have been published in the Journal of Molecular Diagnostics and the Journal of Cancer Genetics, respectively. Very simple process, about 6 ml of CSF can be shipped to the lab overnight at room temperature within 24 hours of collection. Our turnaround time for the test is about 7 to 10 days. Following the launch of these assays, trying to understand the field and the marketplace, we actually did a meta-analysis of the literature to be able to understand what is the level of sensitivity out there for other commercial assays. We looked at input material, the methods that were used, as well as DNA input. And compared to the 4 assays commercially available, the MSK Impact, the UW Oncoplex, the Liquid Trace, and the Summit assay, we found that the Summit, using 20 nanograms of DNA, was a lot more sensitive compared to the other assays out there. So clearly more sensitive and better specificity. Since then, we've published a lot of case reports and research articles demonstrating the clinical utility of both these tests. Recently, we've expanded on the panel. We moved from 32 genes to now doing a comprehensive genomic profile and analysis, looking at 500 plus genes to be able to inform diagnosis for WHO and NCCN guidelines, risk assessment, treatment options, serial monitoring, to track effectiveness of therapy, tumor evolution, as well as support patient selection for clinical trial enrollment. The assay has pretty high limit of detection in the terms of 0.3% variant allelic fraction. We're able to call tumor mutation burden, microsatellite instability, as well as fusions and gene level copy number variants. The analytical sensitivity and specificity of the assay was greater than 90% across all variant types evaluated. And we've got a couple of cases to demonstrate that, yes, indeed, we are able to detect fusions as well as tumor type specific variants within this, within the CSF to inform diagnosis of CNS tumors. And so in summary, what I've shown you is that liquid biopsy has tremendous potential to inform diagnosis and management of cancer. CSF has been identified as a more promising liquid biopsy fluid for brain tumors. And then per CNS guidelines, NGS and CSF is the preferred approach for pathologic workup of CNS tumors. And then the Belay tests have greater than 90% sense and spec. I'd like to thank you all for your attention. I realize this is a virtual presentation and we don't have dedicated time for questions, but I'm happy to address it via email or any other way. Once again, thank you for your attention.
Our next speaker is Dr. Mohammed Shoaib. He earned his master's in molecular oncology and PhD in molecular and cell biology from University of Paris, Sud, France. He then received William Foundation postdoctoral fellowship and joined Professor Sorensen's lab at Brick, University of Denmark, University of Copenhagen in Denmark. He then, next 23 years, he worked on deciphering the role of chromatin structural organization in maintaining genome stability during DNA replication and transcription. In 2001, he joined LUMS as an associate professor in the Department of Life Sciences and established the Epigenome and Genome Integrity Lab. It's a real pleasure to have Dr. Mohammed Shoaib here. He's an excellent researcher who is published in very high impact journals such as Nature Communications, Nucleic Acid Research, Genome Research, and he's also an excellent teacher. So we look forward to his talk. Over to you, Dr. Shoaib.
Thank you very much for the generous introduction, Dr. Faisal. He's my colleague as well, so he's very kind. And thank you very much, Dr. Amina, for inviting me on this auspicious gathering of professionals from oncology, both clinical side and molecular side. And I think this project is also very pertinent because it's at the intersection of clinical oncology and molecular oncology. So what I'm gonna talk about today is an innovative project that we started off a few years ago in collaboration with Shaukat Khanum Hospital, Lahore. And it's a pilot study, and we're working on both breast cancer and head and neck cancer. And what we're trying to achieve is we're taking a different approach for therapy for solid tumors. And it's based on an interdisciplinary approach. It's based on an interdisciplinary team, which includes bioinformaticians, molecular biologists, cell biologists, and clinicians. So I will start my talk will be divided into three parts. So I'll start with the problem statement. I'll tell you why we were motivated to start this project, or why we are taking this approach for breast cancer therapy. And then I'll go into some of the data that we have collected. It's very early days. We just received the data from patients, and we are still analyzing it, so there's not a lot on that. But as a whole, I'll try to explain to you what the approach is. And in the end, I'll talk about a little bit of perspectives. So, sorry for this technical problem. Right. So the fundamental motivation why we were interested in this innovative process, the genome-based personalized therapies for breast cancer, is because of the heterogeneity of the solid tumors. And breast cancer is not the only one. Most of the tumors, they are notorious for heterogeneity. So I'll explain what heterogeneous means is, but I think most of you would already know that just looking at the clinical picture and what people look in the clinic, they look at these three receptors, so ER, PR, and HER2. And I can tell you that this is just the tip of the iceberg. And there's a lot of other mutations or a lot of other factors that actually govern the response to therapy, the prognosis, the development of metastasis, and et cetera, so on and so forth. So what is tumor heterogeneity? So tumor heterogeneity is the biological diversity of tumors. And that biological diversity is based on different features. So you all know that there are a lot of mutations. So genome instability is the hallmark of cancer. Then we have epigenomic alterations. That includes, we just learned about the DNA methylation used as a biomarker. Then there are chromatin accessibility, there are histone PTMs. There is a lot of epigenomic features that govern how tumors behave, and that has an impact on the transcriptomics. And then we have, of course, the proteomic side. So there are post-translational modifications, there are protein stability, protein-protein interactions that may be sometimes independent of the genetic heterogeneity. Then we have metabolic disturbances, and actually a lot of people believe that it's the metabolism that drives, that is the initial driver of the tumors. And then we have the tumor microenvironment. So tumor microenvironment includes the angiogenesis, the infiltration of immune cells into the tumor, and that drives different tumors. But breast cancer, talking about breast cancer, it's considered as a cold tumor. So it doesn't have, so majority of the solid tumors, they are cold tumors with some few exceptions. But especially for breast cancer, there is not a lot of immune infiltration. And that's why most of the breast cancers are usually poor in responding the immunotherapy. So if you look at the tumors, the pointer will मेरे back से right. So these are the different features that I just talked about. So because of this biological diversity, no two patients are alike. So their tumors, breast cancers, they are not the same. They have genetic differences, they have proteomic differences, their gene expression is different, and hence their treatment response is not the same. So for some patients, the standard therapeutic regimes, they will work. On other patients, it will not work. And in some patients, it actually leads to side effects. And all these characteristics, they lead to eventually poor survival or variable survival rates. So this actually motivated us to develop, to think about out-of-the-box solutions. So currently, the current medicine for breast cancer or for generally for the solid tumors revolves around these three regimes. So surgery, chemotherapy, radiotherapy, and in some cases, targeted therapy. But as you all know that most of the target therapies, eventually, there's only a small percentage of patients that can benefit from the targeted therapies. So in breast cancer, there is Thank you. So in breast cancer, there is a new adjuvant chemotherapy nowadays, and that is followed by surgery. But sometimes there is also adjuvant therapy followed by then radiotherapy as well. But still, there are variable rates of therapeutic response. And these variable rates then turn up into a large unmet therapeutic need. And that's why we decided to look at this problem in a different way. So what are the personalized therapy approaches that are existing today? So for example, you can make patient-derived mouse models. You can also make patient-derived cell lines, and then you can test different drugs that are available. You can look at their genetic makeup. You can look at the transcription profile, and you can devise some therapies. These therapies, again, will work on some patients, but then again, there are non-responders. So what to do with these non-responders? So what we are proposing is we're proposing an in silico tumor modeling, and that is followed by derivatization of therapeutic combinations that eventually are tailored to the molecular profiles of these patients. So in this regard, we are looking only at the two of the factors that initially led to the heterogeneity. You remember I gave you a list of different factors. So in this current project, in this current study, we are looking only at the two factors. So the genomic instability, the mutations that the tumors are accumulating, and the gene expression profile. And using these two molecular features, we analyze them in individual patients, and using state-of-the-art software that is developed by our colleague at LUMS, Dr. Safila Chaudhry, and I'll explain a little bit later how it works. So this data can be used to tailor therapies for individual breast cancer patients. So the goal of this project is to develop personalized genomics-based or the tailored therapy to the for the breast cancer patients. So overview of this project, the whole project is, so we take breast cancer patients from samples from Shaukat Khanum. We analyze their genomics profile. We perform whole exome sequencing, RNA sequencing. And what we also do is we make a molecular network. So molecular network, this biomolecular network is an abstraction of the signaling pathways that are working inside the tumor cells. And this biomolecular network is breast cancer specific or breast tissue specific. So once we have the biomolecular network, we can annotate this biomolecular network. So what you are seeing here as circles in this network are basically genes. We call them nodes. And these are interconnected, the way the signaling networks are interconnected. And once we have the genomics data, the exome-seq data, and the RNA-seq data, we annotate this network. And then this is the software Tyson Theater for In Silico Systems Oncology that is developed by Dr. Safila Chaudhry, our colleague. And this performed the in silico modeling of the individual patient's tumor. Once we have the in silico modeling, we have personalized networks. So this network is then once annotated with patient A, will give you a personalized network for the patient A. And likewise, so you can personalize it for any number of patients. Once you have personalized networks, you can treat this network with drugs because these are all genes. You can tell the computer to inhibit one of the gene or few of the genes in combination. You can test different combinations and then see how the network responds. So network gives you an output. You give an input to the network. The input was RNA-seq and exome-seq, and the network gives you an output. That output are actually the cancer cell fates, which I'll explain a little bit later. So some of the therapies will not work again, but then we have a tremendous opportunity to use as many drugs or as many combinations as possible, depending upon how much computation power we have. And then eventually the goal is to arrive at a particular combination that is tailored to a particular patient's genomics profile. So we started this study in 2022, and so far we have collected 109. We have recruited 109 breast cancer patients. So we started this study earlier, but this patient collection was, so the sample collection was started in 2024. And out of those 109 patients, 79 breast cancers were acquired, and we made sure that these patients did not have any previous treatment. So once we had the patient sample, we did two things. So first, we made a tumor cell line, patient-derived cell line. So we dissociated the tissues, we grew them on plates, and then we had patient-derived cell lines. So this is a model that we are going to use to test the therapies, the in silico therapies later on. At the same time, part of the tumor was used for extracting DNA and RNA, and we performed whole exome-seq and mRNA sequencing. So far we have sequenced 20 of these samples, and I'll show you a few of the graphs of the data that we have. It's just to give you an idea what we are looking at. So this is a tumor mutation burden of these 20 patients. The tumor mutation burden is the number of mutations per MB per megabase of the coding genome. So the human genome is about 38 megabases. So this is the number of mutations per megabase of the genome. And you can see that most of the patients, they are between 5 and 11 TMB, but there are these two outliers that have very high tumor mutation burden. That means that these two guys will definitely not respond to the same therapeutic treatment or standard therapeutic treatment. But of course, we are not sure about the light blue ones either, right? Because they will have different type of mutations. This is a so-called oncoplot. So this plot shows you 25 top mutated genes and the type of mutations on the right side in the legend. So we see some of the genes that are frequently mutated that have been previously observed elsewhere in Pakistan as well, in other countries as well, in international databases. But there are also new genes or new mutations that appear in these data sets. We're still analyzing this data set. Another thing that I want to now connect to the heterogeneity. So we looked at a very specific set of genes, the DNA damage response genes. As you know that breast cancers, they are deficient in this particular pathways. So a lot of breast cancers, they have mutations in the genome stability, genome maintenance genes. For example, BRCA1, BRCA2, ATM, ATR, etc. So when we looked at, we collected this data for the DNA repair factors from the repertoire database, and we looked at which patient has which of these DDR gene mutated. And you can see that there is a heterogeneity of, so these are 40 genes here listed here, and different patients have different sets of genes mutated. So it's not really consistent. Only 7 patients had P53 mutated. Only 4 patients out of 20 had BRCA1 mutated. Only 2 patients had BRCA2 mutated. Only 1 patient has PALB2 mutated out of these 20. It's a small data set. I agree with you. So, but still, I mean, this tells you that even for this small pathway that has only 250 genes, there is an enormous heterogeneity. So to address this heterogeneity issue and to make this molecular network, we use different sources. So we wanted to actually create a network that displays or that has all the cell signaling pathways that are working in a breast cancer tissue or breast cancer cell. So we get the data from different databases, from literature, from differentially expressed genes, from therapeutic genes, from drug data, and we connected them using Boolean rules into a network, right? And this network then is then fed into the software Tyson, and this Tyson then creates this network, as you can see. This network has, you can judge from the colors, it has this purple color, which has the inputs, the processing layer, and outputs. I'll explain what it means. So as an example, an input is an EGF, so epidermal growth factor. It's a growth factor, it binds to a receptor, and then it leads to a signaling cascade within the cell. And eventually it leads to a gene expression or some gene expression. So we consider EGF or similar genes or nodes as inputs. So EGF then activates EGFR. So one of the downstream pathways is RAS-MAP kinase pathway. So these genes are all located in the processing layer. Then we have one of the outputs is cyclin D1, right? And cyclin D1 is connected to the proliferation, which we consider as a cell fate. So what Tyson does is it calculates the cell fate propensities, what you are looking on the right side on the plot. So it calculates for a given patient's tumor profile. So by feeding information about their gene expression and the mutational database, we calculate the cell fate propensities. And these cell fate propensities, they are different for different patients, depending upon what is the gene expression profile, what kind of genes are activated or not. So that gives us individualized networks. For each patient, we have an individualized network. And in one of the networks, for example, these 5 genes are mutated, right? So we turn them off or on depending upon what kind of mutation that is. And then what Tyson does is it personalizes it for each individual patient. Now, once the network is personalized, we go to the drug data bank. We look at what kind of drugs are available for all these genes that are present in the network, right? And if we find some of the druggable targets in the network, we then apply those targets in silico. So everything is now happening in silico, still yet happening in silico. And what Tyson does is it gives us cell fates before and after therapy. So we had cell fates before therapy, so it was giving us increased proliferation, decreased apoptosis, et cetera, et cetera. But after drug giving therapy, we see that apoptosis increased, proliferation is decreased, and so on and so forth. So in this way, we can personalize the networks with the therapies, and we can call it a personalized therapeutic network for each patient. Once we have these personalized therapies for each patient, and they can be combination of different therapies, they can be single therapy, chemotherapy plus targeted therapy. We made this patient-derived cell line, which are also a personalized kind of model for that individual patient. We can now apply these drugs, these combinations or single drugs or targeted therapies, whatever came out of the Tyson software. And we use these cell lines to test those therapies, the combinations. And again, I understand it's not the perfect model. It's a 2-dimensional picture of a 3-dimensional tumor. It does not have an immune system, et cetera, no angiogenesis, no tumor microenvironment, nothing. So it has its limitations, and we understand that. So once we test these combinations on the patient-derived cell lines, it gives us some response. Some therapies will respond, some others may not respond. But at least we can now give an informed opinion to the clinician that what kind of therapies may respond better on a particular patient. So we tested this on established breast cancer cell lines. So this is the data for 2 of the cell lines. So MDMB-231, which is a triple negative breast cancer cell line, and KAMA1, which is a luminal type. And we applied the in silico therapy, AC therapy, so doxorubicin and cyclophosphamide on these 2 cell lines. And as you can see from the plots, that the apoptosis post-therapy is going up, the proliferation is almost gone, survival is almost gone, etc. And similarly for the second cell line, KAMA1. But this is again all in silico analysis. So in future, now, since we have the data available for 20 patients, and we have the cell lines available for these patient-derived cell lines available for these patients, we are going to test personalize the networks with the patient's data, and then apply in silico therapy. Once the combinations come out, we can apply those therapies to our breast cancer cohort. We can also expand this study to a larger cohort. So this network has the potential to identify novel combinations. And in turn, we are dedicated or motivated to provide Pakistani breast cancer patients with an innovative, personalized therapy at an affordable price. So finally, I would like to thank my research team. So Faria is my PhD student who has worked most on this project, along with Wajiha and Zara. And Dr. Safiullah Chaudhry, who is our collaborator from the bioinformatics part. So he has designed and made the software Tyson. And his team, Dr. Amir Faisal, is here. So he's our drug discovery guy. So we're very happy to have him. And of course, without the help of the clinic, the Shaukat Khanum, it would never have been possible. So Dr. Rumina Kazi, Dr. Kashif Asgar has moved on. But we started this project together with him and Hassan and Asim. Asim is also sitting here. Thank you. Dr. Farah Rashid, Dr. Saima Faisal from the clinical research department. Thank you very much. And of course, the surgeons. And finally, I would like to thank the funding agencies. So both these projects, head and neck cancer and breast cancer, were funded by HEC and NIH Islamabad. So thank you very much for your attention, and I would like to have any questions that you have.
Okay, excellent, Shoaib, excellent. We'll have time at the end for questions. So those of you who are really have questions on your mind, if you can hold off for another half an hour or so. Shoaib, once again, thank you. So we move on. Our next speaker is Dr. Zishan Ansar Ahmed. And we would have loved to have him here live in front of you, but somehow, as fate would have it, he's in Karachi. And he's going to be delivering his talk from there. I believe it's a recorded talk, or is it a live talk? He's alive. So Zishan, he's an assistant professor and section head of molecular pathology at the Aga Khan University Hospital in Karachi. He's a specialist in molecular pathology and hematopathology, and his work focuses on advancing molecular diagnostics and precision medicine in Pakistan. He has received several prestigious recognitions, including the US CAP Foundation 2023 Mullick Award and the CAP International Fellowship Award in 2023. And he continues to contribute significantly to research, education, and diagnostic innovation in pathology. And this afternoon, the title of his talk is going to be on precision in hematological malignancy practice from genomics to therapeutics. Zishan, over to you. We wish you were
Thank
here.
you. Thank you, Dr. Qureshi, and thank you, everyone. It's a great opportunity to discuss about the precision medicine and practice for hematological disease. And I will be focused on the myeloid leukemia. I would like to thank the organizing committee, Shaukat Khanum, and everyone who is involving and inviting me. So may I have my slide, please?
Do you have your slides or do we have your slides?
I think, or do you want me to share my slide? Okay, I'll share from my side. No issue.
Please, be easier, I think.
I think they have in my. Okay, sorry for delay. Sorry. Am I audible for everyone? Yes. Okay. So as I said, I'll be focused on the myeloid leukemia. Next, please. Next, please. I think it's not.
Zishan, I think it might be easier for you to play
Yeah,
your presentation. Can you do that? Share screen. Can you share your screen?
Yeah.
Hmm. Uh, Zishan, if you don't mind, can we move on with the program and have you on hold, standby?
Yeah.
And
No, just give me, actually, I'm using from another laptop. So it's here. It's here.
You have it?
Yeah, I have it.
Okay, good. Allow.
Oh my God. Mm. Yeah, because I'm using another, laptop from my, secretary, and it's not uploaded. So I already sent my presentation to the secretariat, and if they can, yeah, please. And please move, as I said, next.
After the next talk, we'll be back to you, inshallah. Okay.
All right. Our next speaker now is Dr. Muhammad Usman Rashid, who is a senior research scientist and head of basic sciences research at Shaukat Khanum Memorial Cancer Hospital and Research Center. He earned his MBBS from University of Punjab and did his PhD and postdoctoral training at the German Cancer Research Center, the famous DKFZ. Heidelberg is Fellow of Royal College of Pathologists, London, and Union of International Cancer Control, Switzerland. He has over 24 years of experience in cancer genetics, biomarker discovery, and precision oncology, with more than 100 peer-reviewed publications. His pioneering work in cancer genetics and precision oncology has earned international recognition, and he's dedicated to transferring his expertise to the future generations. He's going to talk about genetic landscape of Pakistani familial breast cancer patients using multi-gene panel testing. Over to you, Dr. Usman.
Assalamu alaikum and good afternoon. Thank you very much, Dr. Amir, for the kind introduction. So I think this talk really fits well after Shoaib, your talk. So maybe just due to this, it just happened. So breast cancer is a clinically, biologically, and as well as epidemiologically heterogeneous disease. It ranks first in both industrialized as well as in developing world. Identifying individuals with an increased risk of breast cancer is an important aspect of modern oncologic practice. Historically, genetic testing primarily relied on sequential single gene testing. With the advent of NGS, multi-gene panel testing has replaced sequential single gene testing. But one of the challenges faced by clinicians is the identification of right candidates for genetic testing, and as well as what are the potential genes with clinical action. So globally, more than 2 million new breast cancer cases are being diagnosed each year, and 500,000 of these women die from this disease. Here you can appreciate a disproportionate number of deaths are reported in low middle income countries from Asia, Africa, and South America. What do we know about breast cancer in Pakistan? Pakistan is the fourth largest population in Asia. October is the breast cancer awareness month, but still there is a lack of breast cancer awareness. On average, there is about 15 months delay for the patient from home to the clinic to present. And for that reason, majority of Pakistani females are diagnosed at an advanced stage. It is estimated that 1 out of 9 Pakistani females will be diagnosed with breast cancer during their lifetimes. And breast cancer accounts for about 1/3 of all female malignancies. About 31,000 new breast cancer cases are being diagnosed each year with the incidence of 35 per 100,000 females. And the mortality is 18.6. And Pakistan has the highest mortality of breast cancer among Asian women. And contrary to the Western world, majority of Pakistani females are diagnosed below 50, and there is increased prevalence of one subtype of breast cancer, which is triple negative. In Pakistan, it is up to 30%, whereas in white population, it ranges from 12 to 20%. So about 80 to 85% of breast cancers are sporadic, having no family history, but about 15 to 20% of all breast cancer cases are familial. And high penetrance and low penetrance genes, moderate penetrance genes account for about 20% each for familial breast cancer. And genome-wide association studies have shown low penetrance genes also contributes up to 18%. So here you see high penetrance and moderate penetrance genes, which are very rare, less than 1%, whereas common low penetrance genes, which are very common, but the risk is less than twofold relative to the general population. The management guidelines exist only for few genes. So there are more than 20,000 genes in our body, but can we just translate all genes for any specific disease? No. So when to consider germline testing for breast cancer, there are several guidelines from different organizations having different age factors and different genes which should be screened. But the main thing is identifying a population-specific and clinically relevant multi-gene panels, which is crucial for any kind of translational research in that specific population. What is a multi-gene panel testing? This is sequencing of multiple genes simultaneously, ranges from 5 to more than 200. And when to consider, choosing the right panel is very important. When more than one gene can explain an inherited cancer syndromes, it could be phenotype directed based on personal or family history to ensure that all relevant genes are included. And for those individuals who are tested negative for a specific syndrome, but personal or family history still suggests inherited contribution. There are benefits, which is cost effective, time effective, with overall short turnaround time as compared to conventional sequential single gene testing. But there's also increased detection rate of pathogenic, likely pathogenic variants. And there is improved management for patients identified with pathogenic, likely pathogenic variants in clinically actionable genes. So there are limitations as well. How many genes should be analyzed? Not all genes are necessarily clinically actionable. What are the expertise of the individual or the lab? Because it includes complex workflow, bioinformatics support, and it adds complexity. Pathogenic, likely pathogenic variant in more than one gene may lead to difficulty for the clinician, for the risk management. And there is also increased likelihood of VUS, variants of unknown significance. And what about the familial testing? Who would finance those relatives to be tested if a pathogenic mutation is identified? So ideally, it should be offered in the context of professional genetic expertise for pre and post-test counseling. The prevalence and spectrum of pathogenic, likely pathogenic variants rely primarily on personal and family history, clinical pathological features like triple negative breast cancer, geographical origin, and ethnic origin. So which gene should be included? So multi-gene panel testing is well established among developed world, but the gene number vary. It depends widely across academic versus commercial labs. And according to the recent ESCO guidelines, breast cancer patients should be tested for these 14 genes. 7 of these are strongly recommended, other 7 are less recommended. Similar 14 gene HBOC core panel has been validated in German HBOC breast cancer patients, but a weak association between additional gene has been reported in a large study of a consortium, more than 100,000 females. So what do we know about multi-gene testing in Asia? Asian comprise about 60% population of the world, with 4.5 billion people are living in Asia, with more than 2,000 plus ethnic groups. But the multi-gene panel testing is underused due to resource challenges and due to poor understanding. Interpretation of identified variants leads to increased frequency of variant of unknown significance. Diversity matters, but why this is the case? Because more than 86%, you can appreciate, 86% of genetic studies have been primarily conducted among European population. So among Asians, high-risk breast cancer patients in these different countries, from 100 to more than 4,000 samples have been screened for genes ranging from 20 to more than 100, with the frequency of pathogenic, likely pathogenic variant ranging from 7 to 30%. So beyond 14 gene panel testing, there are no additional variants identified among studies from Jordan and China, and no single gene exceeded a frequency of more than 1 even after testing up to 94 genes. So limited clinical utility of extended panel has been noted. Over a period of last 20 years, we have conducted sequential single gene testing and identified pathogenic, likely pathogenic variants in these different genes, which accounts for about 40% of familial breast cancer from Pakistan. So multi-gene panel testing in Pakistan is very limited. So far only 2 studies have been conducted, NGS-based multi-gene panel testing in Pakistan. Both studies are from Karachi. First study based on 84 sporadic breast tumors having no family history. 27 genes were screened with the frequency of 12%. Second study was also performed from Karachi, Aga Khan, Salman Kirmani, and Fiza. So they included 273 high-risk cases, including 167 familial breast cancer and several other cancers with it. 27 or 37 genes were screened, and the frequency of pathogenic, likely pathogenic variant was 22%. But there were more reliance on the foreign testing because there is a limited availability of NGS expertise in Pakistan. So there are no national guidelines for genetic testing in familial breast cancer. So which gene should be included? So for that reason, identifying a Pakistan-specific clinically relevant multi-gene panel is crucial for the effective implementation of testing into clinical practice. So we conducted a study to investigate the diagnostic yield of 14 genes panel using NGS in Pakistani familial genetically enriched cases who were tested negative for pathogenic variants in different genes. So of these, they were negative for BRCA1, BRCA2, PAL2, TP53, RAD51C, and RAD51D. So 263 families were recruited in a period of June 2001 till August 2015. So they were stratified into 3 different groups. All cases were pathologically confirmed with breast cancer. It included 80 cases with F2BC, including at least 2 breast cancer, at least 1 diagnosed below 50, or F3B. 3BC group included 130 cases having 3 or more breast cancers in that family, at least 1 diagnosed below 50, or FBOC group, which were 53, having a family history of 2 or more breast cancer plus 1 ovarian cancer. 103 cases were excluded because they were harboring BRCA1, BRCA2 mutations, and only 160 cases were screened for these 14 genes. So NGS was performed at Shaukat Khanum Memorial Cancer Hospital. These are different steps. Illumina MiSeq was used with the coverage of 100% at more than 50x depth. So variant annotation and classification was performed, and 5-tier classification was used as per American College of Medical Genetics and Genomics. And further refinement was done by gene-specific guidelines available for these 6 genes, and ClinVar database was also accessed. And there was an independent review by expert curator and clinical geneticists. And on top of that, all mutations identified were validated by Sanger sequencing. What we found, 29 out of 160 cases were identified with likely pathogenic variant with a frequency of 18%, whereas VUS was identified in the frequency of 13.1. Here you see the distribution of likely pathogenic variants. So BRCA1 was the most common with a frequency of 8.8. Second was BRCA2, 3.8. Third was TP53, 1.3% in CHEK2 and PALP2, and 0.6% each for ATM and RAD51C. So here you see the germline likely pathogenic variants. Among these, 2 BRCA1 and 1 PALP2 mutation was recurrent, identified in unrelated families. All were reported in ClinVar except 1 BRCA2 mutations. No likely pathogenic variants were identified only in these 7 genes mentioned here. So here you see the characteristics of 29 families harboring likely pathogenic variants. So among these, so you can see the index patient is mentioned with asterisk. There are several other cancers were also reported. Here you see the geographic or ethnic origin. And breast and ovarian cancer families were only identified in BRCA1 and 1 in BRCA2. For the remaining 5 genes, they were only identified among familial breast cancer only cases. Here you see the overall frequency of likely pathogenic variant using NGS. So overall frequency was 18.1, but for BRCA1, it was 8.8. For BRCA2, 3.8, and 1.9 for TP53, and 1.3 for CHEK2 and PALP2, and 0.6 for ATM, and for RAD51C, also 0.6. Of the identified mutations, BRCA1 and 2 accounted for about 70% of all mutations. When we stratified our cases based on triple negative versus non-triple negative cases, you can see that among triple negative cases, the frequency was even higher, 22.5, and mutations were only identified among BRCA1, PALP2, and RAD51C. Whereas in non-triple negative, almost it was identified in 6 out of 7 genes. So based on that, clinicians could suggest which gene should be screened for triple negative breast cancer cases. So when we combined our data, the current one and also previous conventional, less sensitive technique used on the same cohort, the cumulative prevalence increased to 50%. 132 out of 263 patients were identified with these mutations, and there was BRCA1 predominance. It was even higher to 93%. 123 out of 132 were identified with BRCA1 mutation. And of the identified BRCA1 mutation, about 60% were recurrent, and we have reported these are founder mutation, Pakistan specific. So overall, among families with 2 breast cancer cases, the frequency was 38.8. It increased to 48.5 among families with at least 3 or more breast cancer cases. And among breast and ovarian cancer families, it increased to 71.7. This is quite significant. So here you see the clinical and pathological characteristics. So there was a significant difference in the age of disease onset. Among BRCA carriers, the mean age was 35.9, whereas among non-carriers, it was 44.8. And there was a significant difference was found among laterality, histology, and tumor grade, and ERPR receptors, and TNBC, and the familial group. So here you can see the NCCN recommendation for these 7 genes: risk-reducing mastectomy or risk-reducing self-angioprectomy, and PARP inhibitors. This is the FDA-approved drug available in the market for the clinicians for BRCA1, BRCA2, and PALB2 carriers identified with these mutations or pathogenic variants in these genes. So VUS were also identified in different genes. So what are the strengths and limitations? This is from the single center. So the uniform criteria was used for the pathological classification of patients. Ethnic diversity. Shaukat Khanum is a tertiary care cancer center where 75% of patients are getting free treatment coming from all over the country. And confirmation of all variants by Sanger sequencing. The limitations that copy number variants and large genomic rearrangements beyond BRCA1-2 were not screened. What are the clinical impacts for the clinicians? This is the first nation-led large-scale multi-gene panel testing, specifically in familial breast cancer using NGS, where 7 genes accounted for about 50% of pathogenic variants. It provides a population-specific genetic profile and proposes a cost-effective first-year genetic testing strategy. And it could inform national clinical guidelines and reduce reliance on foreign testing and improve access to precision oncology among Pakistani females. So in summary, these are the 7 genes, and there is the predominance of BRCA1 and 2. And this is the paper published 2 months before in International Journal of Cancer. So this is the landscape, our journey in the last 24 years, showing the credibility in genomic research. At the end, I'm thankful to the study participants, study coordinator, especially Dr. Saima Faisal for her active involvement in the recruitment, and the molecular work by Dr. Noor Muhammad. He is a postdoctoral fellow, and Shumaila. They have actively contributed to this NGS study, and Humaira Naimi for the preliminary BRCA1 screening, and my collaborator Ute Haman from German Cancer Research Center. And obviously, it was not possible without the support of our management. Thank you very much for your attention.
Okay, thank you very much, Usman. Questions later on, but we need to go back to Dr. Zishan at Khan University.
Thank you so much. Without
Zishan, are
delaying,
you-
yeah. So right now the presentation with the secretariat, and I'll just go with them as I will say the next. So I already said my topic is the precision and hematological malignancy practice from genomics to therapeutics, and I will be focused on myeloid neoplasm. Next. Next. I have nothing to disclose. Next. I will be discuss the biomarker and recommendation for characterize the myeloid neoplasm according to the WHO. I'll be discuss the molecular genomic testing for hematological malignancy, their advantages and the application, and the last, the challenges and the limitation of genomic testing in hematological malignancy. Next. So actually, this is the basics why we need to actually do the genomic analysis. The genomic analysis of any kind of cancer, especially in molecular cancer, to know the diagnosis, characterize the disease, classify the disease, the prognosis of the disease, and the important thing currently, since last 2 decades, the predictiveness of the disease, to know the actionable marker which already discussed in previous sessions, and the last to follow up the disease by measurable residual disease to monitor the disease next. So this is a very interesting, editorial by histopathology, and they actually want to know why the WHO classification is used. So what they identify that the majority of the uses is for classification and the diagnosis, and the other two, contribution by the research and the development. And what the most common thing they identify is the molecular pathology itself come as a new discipline which is come in all discipline, means the top second, or you can say in the future it will come as a top discipline in all healthcare discipline. Next. And this is another paper that shows that the all healthcare revolution, the molecular discipline is emerging. Why? Because it's breaking the discoveries through the different molecular marker, which is used as a targetable marker and the actionable biomarker. And the groundbreaking technology is only two, the Sanger sequencing and the PCR. And I will explain it why it's two. Next. So this is about the myeloid neoplasm. So if you gone through the myeloid neoplasm, it's basically in the two extremities. One is the acute leukemia, acute myeloid leukemia, where the blast percentage is higher, around 22%, or it's basically immature clonal proliferation of hematopoietic stem cells. While you come to the myeloid leukemia, the chronic myeloid leukemia, or the chronic myeloproliferative neoplasm, it's basically the clonal hematopoietic proliferation of the mature cells. And there is a one entity in between two, is the pre-malignant or the some atypical or the mixed subtype of the myeloproliferative neoplasm. But the important thing about this slide, that since the classification and the discoveries comes and updated, the importance of these all genetic marker comes as a for the diagnosis, prognosis, and the targetable for the diseases. And if you go through, we focus on the BCR ABL, which basically rearrangement between the ABL present on the chromosome 9 Q arm and the BCL22. So it's basically the ABL which normally function in hematopoietic to control the proliferation, control the differentiation, control the maturation. It's become uncontrolled because the BCR comes to the ABL to fuse it. And ABL basically is a proto-oncogene. It's a drive the clone. So it's a main driver clone for different leukemia, but the most commonly in as a pathogenic diagnostic therapeutic marker for the chronic myeloid leukemia. And why it's basically more, producing a good outcome comparative to other leukemias, because the transcript in the chronic myeloid leukemia, which is P210, is 95% is basically identified compared to other transcript, which is P190, means identify in AML, ALL, and the mixed phenotype. And the TKI is more competent with the P210. So that's why the revolution comes from the tyrosine kinase inhibitor for more in the P210 transcript than the other transcript. But the other factor are for the other, leukemias, that it's a multi-heterogeneous disease. And in the BCR-ABL, usually in the chronic myeloid leukemia is basically derived from the one clone. Next. So because of the its the TKI effect and its biology or pathophysiology, within a year you will achieve the milestone and almost eliminate the clone up till the the low level of identification, or the if you can identify means understand the log reduction within a 12 to 18 years is come 4.5, which is equals to 0.0032 percentage of BCR for those who are favorable. And in new literature, they said it's come up to the 5 log reduction, which is equal to 0.0001 percentage of the BCR ABL comparative to ABL gene for the quantitative analysis. Next. And this is the one study has done that they actually done 20 years study to identify the burden of disease, and the second, the disability adjusted life year with the age standardized. So what they found that the since they started the use of TKI, and it totally replaced the conventional therapy and the transplant therapy, the disability chance is much lesser than the the other therapies, and the disease related disabilities also. So this is a revolution for the TKI. Similarly, non-BCR-ABL myeloproliferative neoplasm also identify a different biomarker, which is the common one is the JAK2, calreticulin, KIT, and the CMPL. But for the non-typical uh, myeloproliferative like CMML, GMML, and the MDS-MPN, there is biomarker like AXL, TAT, uh, JAK2, and the 1 inhibitor, and these basically emerging actionable prognostic marker, which is available for new targetable agent with the promising outcome. Next. So come to the acute myeloid leukemia. So as I said, since 2008, the WHO is starting to reclassify the disease, especially the acute myeloid leukemia, and they give the one entity of the AML, which is the genetic defined abnormality of AML, which has the specific biomarker or the genetic change which define the acute myeloid leukemia without even having a percentage of blast, even it's less than 10, less than 20. And they not actually basically diagnose the disease, they actually also the actionable target for the different targetable agent, which is you can monitor like the different transcript, NPM, IDH1, JAK2, KIT, BRAF, and the FLT3. The FLT3 is the most aggressive marker, and due to this, the disease relapse is higher. Next. So these are the outcome. Because of these marker, the outcome of the acute myeloid leukemia, which is very aggressive myeloid leukemia, or the in all leukemias, the outcome comparative to the conventional therapy or the chemotherapy with the target therapy is better than since last decade. Thank you. Next. Similarly, the acute myeloid leukemia for the pediatric group is much more poor prognosis than the ALL. But because of these emerging marker, especially in even the high risk, the CBF2, GLI, UBTF, even the MLL and the NP NUP8 rearrangement is the new emerging marker, which is targetable, and the emerging target treatment give the compromising or the better outcome. Next. So with these, emerging marker, the conventional or the you can say the therapy, the conventional chemotherapy is totally replaced by the personalized medicine. And the example are the most successful result is for the chronic myeloid leukemia by the BCR-ABL inhibitor, and the other myeloid leukemia, especially the one emergency leukemia, is acute promyelocytic leukemia, and the other leukemias like the recurrent translocation, which give the favorable result for the acute myeloid leukemia. Next. I have add the one slide because the lymphoid group for Hodgkin and non-Hodgkin is a very huge group. So, but the diffuse large B cell leukemia is the one of the common entity of the non-Hodgkin lymphoma. So before they were categorized according to the morphology and the immunohistochemistry, but since the biomarker and the genetic feature is available, they also working on targetable therapy for these marker, and the outcome will be seen good comparative to the chemotherapy. Next. So these are our publication which we publish. Some of them are the published abstract which we present in the Society of Hematology and Oncology in MD Anderson. Some of them are the largest study group, especially the FLT3 in AML and the chromosome abnormality in AML, which we have done around 300 or 400 group study. But otherwise they are the case studies and the case reports. Next. So the challenge for genomic testing is basically the quality and the quantity of the nucleic acid, either as DNA or RNA, and the tumor fraction. Next. And second thing, the type of sample. So ideally in hematological disease, we actually either need a blood or the bone marrow. We don't save these kind of viable tissue for a long time. So if we are doing a chromosomal study, ideally we need to have the live cells. But for the genomic study, you can extract the DNA, and you can save for the genomic study. Next. And second challenge is the viability and the cellularity of the specimen, the type, the tumor fraction quality, and the processing of the tumor or the specimen. And the last is the ideal process, that while you are collecting the bone marrow or the blood, you have to send it for morphology, for immunohistochemistry, and similarly same time you have sent for the genetic marker, so the time will be saved for the patient management. Next. So if we have the highest cellular content of the specimen, you will have the high nuclear or the DNA yield. But if you have the hypoplastic specimen, you will not have the sufficient or the good quality of yield of the DNA or RNA. Next. So ideally, next. So ideally, we have to actually go through and review the specimen quality, and as much as I said, the nuclear cellularity is directly proportional to the DNA sufficiency or the quality and the quantity. So there are the different genomics tool available for diagnostic of different malignancy, hematological malignancy. Next. So if the chromosomal analysis, as I said earlier, if you are doing it, so you can do either a conventional or the interphases studies. So you can do the fish and the other study. For conventional the karyotyping, we have to have the bone marrow for precursor hematological cells. But for genomic study or the DNA or the gene study, there are different the genetic or the different diagnostic tool available at the DNA or the RNA level, which is either if you are just targeting the variant, you can do by the PCR, but if you are sequencing it, you have the Sanger or the NGS. So this is next. This is how basically the genomic testing is evolving from chromosomal analysis to the molecular analysis, and it give you the overall picture if you are targeting or screening the whole chromosome. But if you are just precising the some target, either a sequencing PCR or the fish, so you will be target of gene of the interest or sequence of the the gene which is your interest actually. Next. So as you know, my previous colleague also discussed the tumor mutation burden. So usually tumor mutation burden is done for the solid tumor, because the now the if that as you know the tumor mutation by the 10 megabase is higher, so it's ideal also for the immunotherapy. It is still not you know established for the hematological cancer, but if the tumor burden is higher, is directly proportional to the detection of the mutation. So similarly, as the tumor is grow, so you can easily pick and follow its the beginning level as a for follow-up monitoring for the measurable disease, but for early detection by the genetic studies. Next. So there are different sequencing option. Uh, you know better than me. If you are targeting the whole gene, whole DNA, whole DNA is a genome. If you are just focusing for the exon or the coding region, the exome sequencing. But in diagnostic setup, the ideal one is the targeted panel sequencing. And I will tell you why. Next. So generally, the whole genome and the whole exome sequencing are those in the clinical setup are for those where the primary or the diagnosis or the primary confirmation of the disease is not known, or either it's an inconclusive diagnosis, or also for the research purpose. But there is now the panel available for the myeloid, lymphoid, and the acute and the chronic leukemias or the lymphomas, which is more cost effective and have a high accuracy. Next. So there is one study have done just published that they have the two cohort. One they selected who have more than 300 gene panel, and one they have the less than 100 gene panel. But the availability of sample is around less than 50%. But what they actually their objective is to find out the molecular base recommended therapies marker. So in the the larger panel, they have identified the more gene which is targetable than the small gene panel. But you know for these kind of study, they have to do large group of study or the large number of patient studies. Next.
Dr. Zishan, can you wrap up? 1 minute.
Sure. Okay. Next. So it give it early detection, and it's a time you can monitor and select the therapy. Next. And the limitation is already discussed, that you can miss the large deletion and insertion. The conclusion is basically the molecular hematology is transformed for diagnosis, prognosis, and it gives the selection of the therapies. And the continued innovation, it give the different kind of information. So NGS is changing the process of molecular testing in these kind of patient, and with time it will be possible that it's come as a routine diagnostic in our setup also. So we have a different hematological panel available, and the different PCR based testing for the myeloid and the lymphoid leukemias. Next. And we also providing a packages form by the NGS based testing. Thank you for your attention, and I'm available for the questions.
Thank you very much, Dr. Zishan. Thank you very much. So we are at that juncture. We have about 10 minutes left in this session. And this is the Q&A. This is the Q&A part of the session. So for this, I'd like to request Dr. Usman, Dr. Schwab, Dr. Zishan, can you stay online, please? Usman, come up here. You guys can have these chairs. And there's a mic, somebody with a mic. So if you have a question, by all means, raise your hand and go ahead and ask. Question? Okay, please.
Assalamu alaikum. My name is Fiza Akbar. I'm working as a genetic counselor and a genomics researcher at AKU. Wonderful talks by all the speakers. Just had a few questions from Dr. Usman's incredible work. So Dr. Usman, you had a really stringent criteria in terms of the recruitment that you did for your study. And the diagnostic yield was about 50%. So my question to you is, I've got a couple of questions. The first is, how do you plan on sequencing the rest of the 50%? Because they still are very convincing cases for further analysis. So do you have any thing planned out beyond multi-cancer gene panel? And for the 14% VUS that you had received, were you able to do any segregation analysis to see if those variants of uncertain significance were actually more interesting or disease causing?
Thank you very much. Excellent question. So first question that you mentioned, that 50% prevalence we have identified in our genetically enriched cases. Yes. So these are the genes which are clinically actionables. So means these are the genes which would help a clinician sitting in the clinic so that they could plan the strategy and tailor it. For the remaining 50%, yes, so there are still unknown genes. Like I have mentioned, that copy number variants and large genomic rearrangements. We did screen for BRCA1 and 2. We don't know so about other genes. Among these 7 genes, I would like to screen those ones first, which are in the pipeline. And the second question you mentioned about the co-segregation of VUS, the major thing of VUS in Asian is, I have mentioned clearly, because there's a limited data. There are no Pakistan-wide genome. For that reason, variants identified, so which are not identified in the West, so they just declare it as VUS. So we have to do the functional assays and the co-segregation analysis and the pathology of the patient. But do all patients have the strong family history? Are those individuals alive? So this is the question. But definitely, so that will be screened.
What we had about 50% VUSs, but we use NCCN criteria, which means that we are testing a lot more. So which is exactly why I had that question, that probably you have the opportunity to do a lot of downstream work.
Yeah, because we have the access, the comprehensive data, and the patients and the families are from Shaukat Khanum, so we have the access, but not like you are the genetic counselor, you know that not every family is willing for the genetic testing. So there are so many family dynamics are different for an educated person or uneducated. It varies from family to family, but definitely. But the interesting thing we noted that in VUS, ATM was the most common. And this is also in accordance to the West. So they have also identified, and it's a big gene, as you have mentioned, that more than 61 exons or something like this. So these are the things, but it's a long way to go. Yeah, we will do it. Thank you.
Dr. Natasha.
Thank you. Thank you very much. Natasha Anwar from AKU. Thank you so much, both of you. Great talks. Dr. Shoaib, just a quick question. Really exciting work that you're doing at LUMS. And if I understood it correctly, the first part of the work was actually tumor sequencing. And those are the sequences that you push through your that Tyson, yes, Tyson pipeline. So you were specifically focusing on the missense variants that you identified. I was just wondering, does the model have the capacity to explore VUSs, or are you just specifically right now looking at the proteins that are actually not functioning, and then you have that pathway which is then investigated through the Tyson pipeline?
Right. Thank you, Natasha. So I think it's an excellent point, and we are just beginning with this project. And like I mentioned, it's a pilot project. So if I give you a brief idea about what Tyson does is, so it's a low resolution profiling of mutations. So what you are talking about is a very high resolution. You look at the mutation type, whether it's a missense, it's a frame shift, it's a point deletion, it's an insertion. So at this point, we're not bothered by the specifics of a mutation. But in future, that can be done. So in parallel, we have another project where we have developed a CRISPR-based assay for functional characterization of the variants of uncertain significance. And actually, we have written a grant over it. And I just heard about Dr. Usman. He's also interested in breast cancer variants of uncertain significance. And we are already working with Dr. Romina. So I think if you are also interested, we can work together and we can explore this WUS and characterize them in future. So at the moment, just to answer your question, at the moment, in Tyson, we do not go as deep as an individual mutation. We just look at what is reported about a particular gene, if it's mutated, and the mutation that we are getting, like missense mutation, if it's pathogenic or not. We just use that information that is available. For WUS, we have other projects going on.
Any other questions?
My name is Dr. Naeem. I'm from Punjab University.
Can you speak up a little bit?
Okay. So my name is Dr. Naeem and I'm from the Punjab University. So I have one quick question to Dr. Shoaib. That I just want to know that how the drug discovery part is actually working in the Tyson, that how you are going to discover the drugs from the databases once you have identified the mutations.
Right. So what Tyson does is it models an individual person's tumor. It's an abstraction of a tumor. Why I call it abstraction? Because we are modeling only, currently the network has only 3,500 genes out of the 20,000 genes that are, available in human genome. So we're working only with those 3,500 genes. They are interconnected. And what Tyson does is it creates this network, and then using statistical mechanics, which I also personally do not completely understand. So our bioinformatics colleagues, they do that work. My PhD student, actually, she has also learned a lot about it. But what I understand is using these Boolean rules. So the network is created using Boolean rules. So if there are two genes, we have written a rule how they are connected with each other. If they are activating, one is activating the other, or inhibiting the other, or how are they connected? So all these rules are written. There are 10,000 or 12,000 lines of rules written by Faria and my PhD student. And these rules are then used by Tyson to make that network. So once the network is made and the rules are in place, now you can take down one of the genes in the connected node. What happens is everything that is downstream will be affected because it's a connected network, right? So if you take out one of the nodes, everything that is downstream will have an impact of this inhibition. So you can use druggable targets. So you can you can fix mutations that are found in patients' exome sequencing profile. And if these mutations, for example, if they are in tumor suppressors, you can turn off the gene in silico. And what happens is whatever is downstream that gene, all the nodes that are downstream, they will have an impact, although a slight impact, but they will have an impact of that turning off or turning on. For example, if there is a mutation in PI3 kinase, it's an oncogene, there are activating mutations, you can turn it on and its effect will be, so downstream nodes will be impacted. And eventually what we observe is those cell fate propensities. So the cell fate, for example, PI3 kinase is activated, we should see an increase in proliferation, right? If AKT is activated, so we should see an increase in survival. So it's something like that. So this is how Tyson works, and it's an in silico environment. It's a network model, and once you personalized it with the patient data, you can then play with it. You can turn off any druggable target and then look at the propensities. And if the propensities are going towards normal, so if the cancer propensity, like proliferation is very high in cancer, but after those druggable targets, when you turn them off, you see the propensities are going towards normal. So that would mean that this therapy is actually working.
Sir, is this possible through the Tyson to identify the new drugs for the drug targets, or is it just for the repurposing studies that already drugs that are available in FDA database, they are only going to be identified?
Yes. So it definitely has the potential to identify new targets and actually to identify synthetic lethal targets. So this is our actually next work plan that so at this moment, we're looking only at the druggable targets that are already approved. So we want to repurpose them. We're not interested in looking at identifying new drugs, but it certainly has the potential because you have this network, and out of those 3,500 genes, you can use any gene for a target, because if it's a connected network, then taking out one gene or inhibiting or turning off one of the nodes or one of the genes, it will eventually have an impact. And you can combine different nodes, different genes, combined inactivation or activation. You can look at the cell fate propensities. So definitely it has the potential to identify new druggable targets as well. But as you know very well that not all proteins, they are druggable, right? So but in theory, yes, it's possible.
Hang on.
We can discuss it later in person.
I've got a quick question. I'm the boss around here, so I can slip in a question. Oh, can you make do with RNA-seq only? You don't need the genomic sequence?
So you're asking if we can do this only with the expression data?
Yeah.
Well, it has a limited potential. Even with the genomics data, it has its own limitations because the tumor heterogeneity, it comes from the diverse aspects, from epigenome, from proteins, from metabolome. So the less information you have, the less informative the model will be.
Ali.
Thank you, sir. Thank you, Dr. Shoaib and Dr. Usman, for the very wonderful presentation. So firstly, Dr. Shoaib, I would like to know that does your software or the algorithm that you have just described has the potential to let us know about that which of the following acquired mutation will be across the course of treatment? Does it has the potential to describe or predict the impact of mutations that are going to be acquired?
So not precisely the impact of a particular mutation, but the impact of the gene can be predicted. So an impact of gene, for example, you can turn it off or you can turn it on. At the moment, it's like I said, it's low resolution, but we can enhance it. We can go into, so for example, at the moment, we're trying to inform the model that look at the cosmic database get all the pathogenic mutations, and then looking at our patient's data, matches what are the pathogenic mutations, and then in light of those pathogenic mutations, turn off or turn on the gene. So that is possible. At the moment, we're not doing that. It's in early stages. So we're trying to establish proof of concept.
Okay, thank you. And my next question is to Dr. Muhammad Usman Rashid. Sir, have you encountered any of the intronic and the mosaic variants among your population study cohort?
So we got the panels, we got it customized by the twist panels, and they have included the complete coding sequence up till 20 nucleotides of the intronic region, which was included. And that region I can comment. Beyond that, deep intronic region, so that has a little influence. We haven't screened those ones.
All right, sir. So
Any other questions? Okay.
So I have a question for Dr. Rashid. It's a very interesting study you presented.
Thank you.
I think given the difference in sample size, your findings are probably not very different from those very large studies that have been done in populations of European ancestry. I know it's not something you've done, but I wonder about SNP analysis, because that might look quite different.
Yeah, that's a good question. For the SNPs, so we plan for our Pakistani population to do the SNP analysis to select. There are 313, there are Breast Cancer Association Consortium studies and SIMBA Consortium studies in which we are a part of those studies. So, but the major genes and the SNPs identified, so in single gene screening, so there are unique changes we have identified in this population, which are not described. These are the novel changes. Yeah. So we do plan for the comprehensive, for those risk prediction scores to be used for the screening of those potential, because not necessarily that gene like 5382 insertion C mutation, the most common one, which is not even found once in Pakistani population. So this is one thing. And there are also check 1100 del C mutation, which is very common in European population, but we haven't identified. But we did identify other SNPs and pathogenic mutations in those genes. So those genes have contribution, but with different variants.
Okay, we've run out of time. I promised you this was going to be a great session, and it was, right? Thank you. Thanks to all the speakers. I want to thank the audience for staying here this late. I'm sure there would have been more questions, but really, we don't have time. You can always meet up with the presenters afterwards. So Dr. Zishan, thank you. You're still there?
Thank you. Thank you. Yeah, I'm
Okay.
here.
Thank you. And
Thank you,
Dr.
everyone.
Schwab, Dr. Usman, heartfelt thanks. And Dr. Honey Reddy, I think she delivered a great talk. And, you know, sometimes we watch movies and there are take-home messages. We don't need to interact with people sometimes. I thought her talk was very good. And I think some of the messages in this session have been relayed to you, from liquid biopsies to some of the challenges through which you can make things personalized. This is what we have to deal with as a group. So thanks once again. Let's give them a applause. And with that, I'll close the session on behalf of you also. He's not my co-chair, he is the other chair. So thank you.
So thank you, everybody. We'll officially conclude the proceedings for today's session. And before we leave the hall, please, I would really like everybody to applaud Dr. Amina for not losing her cool when we lost the slides and for keeping calm. So that was great on her part. So hopefully we'll see you tomorrow. Please follow the agenda for tomorrow's sessions, and we'll hope happily have you here tomorrow. Right? Thank you. Thank you.